Oh, that looks terrible. Hello?
Yeah, we can hear you.
Okay, good. Oh, thanks. All right. Still in the process of transitioning from applied stats. But let me get my act together here, and we can proceed. How are you guys doing? I haven't checked in on your mental health in a while, except what comes up in lab. You guys gonna make it to the end of the semester? Elizabeth is smiling, and a smile that makes me think maybe not.
Well, I'm gonna try. God damn it.
Okay, all right, good enough. How are the rest of you guys doing?
Not great, but not terrible.
Who was that?
Quinn.
Quinn. Okay. What's not great and what's not terrible?
Just a lot going on.
Mm-hmm.
Yeah, but we're almost done, so.
3 more weeks of class time, yeah. It's creeping up on us. Speaking of which, this is that time in the spring semester when things start to get very busy. So I just want to remind you that this week is an altered schedule. So Friday is undergraduate colloquium. Kelsey and Elizabeth are both presenting at colloquium. So hopefully you've logged on and looked at the colloquium schedule. It's posted on the Duke colloquium page, which is on the main webpage. There's also a link to it on the Jewel Central in the View from the Hill. There's a little blurb about it in there. You can log on to that. There's a PDF program, and in the PDF program are links to the Zoom meetings, which include all of the oral presentations and also all of the poster presentations. The oral presentations will go on during sessions with 3 oral presentations per session, moderated by a faculty member, and then the poster presentations are each individual student kind of running their own Zoom meeting that you can go and join at any time during the time that is scheduled for posters. So we will have class this week on Monday, Wednesday, and Thursday. There is no lab this week because Thursday is on a Friday schedule, and so we will not meet on Friday because we will all be participating in the Duke colloquium. Those of you interested in medical school, the speaker for the Duke colloquium, normally at colloquium there is a luncheon for the people who present that nobody else gets invited to except the people who present. But because this year there is no luncheon, we decided to still go ahead and have a speaker. But the nice thing about it is the speaker is available to anyone on campus. So there's also a Zoom link for that speaker. It turns out this is the 20th anniversary of the first Duke Colloquium. And the speaker that we have this year, rather than it being a faculty member, which is who normally does the talk, we got one of the first people who presented at the first Duke Colloquium to come back and speak to the campus. The guy's name is Dr. Kirk Mccullough. He is an orthopedic surgeon who practices here in Kansas City after having gone to, I think, Vanderbilt, if I recall correctly, for medical school. And he is an orthopedic surgeon. He's also team doctor, sports orthopedic surgeon for the Kansas City Chiefs, Sporting KC, Kansas City Ballet. He does a lot of consulting with many of our athletic performing organizations here in Kansas City. He was doing undergraduate research his senior year at this time of year, just like you guys were. So it'll be interesting to kind of hear his perspectives on kind of what he's been doing for the last 20 years. He graduated in 2001, so he's been out from college for 20 years. So that might be of interest to you guys in particular. I talked to him on the phone just last night, actually, about his talk. So yeah, so that's going to be Friday. So lecture Monday, Wednesday, and Thursday, and then colloquium on Friday. Questions about schedule? All right. I have signed up for my second COVID-19 shot on the 27th. Hopefully you guys have also. If you haven't gotten your first shot, you definitely need to get that done. I've heard nothing about whether they're going to require vaccinations in order to return to campus in the fall. This doesn't affect any of you guys because you guys are all graduating, hopefully. You guys are all graduating, right? Yes, get out of here. We don't want to see you here next fall. You don't want to be here next fall. If you haven't gotten your first vaccine shot, then definitely do that. And if you did get the first one, make sure that you get scheduled for the second one. It looks like it's going to be the same procedure as the last time down at the Mabee Center. You should have gotten a link via email to sign up. Did you guys get a request for signing up for those things? Okay, good. Because I did not get one. I had to steal somebody else's link in order to get signed up. Luckily, they're not specific to a person like they are in some cases. I heard from a guy that I know at University of California, Dominguez Hills. It turns out that the University of California system is not going back to in-person teaching in the fall. And there are a variety of reasons. One of the reasons is that within the University of California system as a whole, there is a much larger student body. There is a lot of people who live not on campus, so many of these campuses are not residential campuses. They're more urban campuses with people living at home or living in apartments. And so it's a lot more difficult to sort of contain the student body than it is at a residential institution like ours. And so they just don't feel that they can, even with rates of vaccination going up, they don't feel that they can safely start school in the fall with 100% in-person classes. So they're going through and doing an evaluation of what courses really need to be on campus and in person, what courses can be effectively done, remotely, et cetera. So they hope to be at about 50%. So different campuses are taking different approaches. The plan here is to be back in full swing. So you guys won't be here, but you'll be going on to whatever you're doing after this, and they will probably appreciate you being fully vaccinated wherever you go after you leave here. So pay attention to that coming down the pike. That's a Tuesday, if I recall correctly. All right. Okay. we were talking about eggs. So we're going through reptile reproduction and life histories. And so we began talking about different forms of eggs, from parchment shelled eggs to calcareous shelled eggs. And these differences in egg structure basically have to do with whether or not there's a mineral layer added to the egg, or and the degree to which that mineral layer is thickened, and also the shape of the mineral layer as to whether it's in these kind of rectangular blocks that make it more rigid, or if it's in non-rectangular blocks that allow the eggshell to flex somewhat. And so different groups have different kinds of eggshells, and it is to a certain degree phylogenetically determined. So for example, within the lizards that have rigid shells, this is primarily the geckos, but it's just one subset of the gecko family, of the Geckonidae. The eublafarids basically lay parchment shelled, and all the other geckos lay rigid shelled eggs. And so there is quite a lot of phylogenetic canalization or phylogenetic inflexibility in terms of the type of egg that you lay. You tend to lay eggs that are similar to other species that you're phylogenetically closely related to, and that shouldn't come as a surprise to you guys at this point in your careers. So we talked about that last time, and we didn't get into oviparity versus viviparity, but there are plenty of viviparous lizards, and your book talks about kind of what the tendencies are for determining whether or not a species has evolved to viviparity. But this is kind of how it's arranged structurally. What you generally see in an oviparous egg is you
Dr.
see.
Kowinski, you're not sharing your screen right now.
Oh, crud, I'm not sharing my screen. Thanks for telling me that. All right, am I now?
Yes.
Okay, thanks. Yeah. Spent too much time in preamble. Okay, so here you have two different eggs. These are schematics from your textbook. On the left-hand side is a oviparous situation. You have a well-developed yolk that is separate from the allantoic cavity. So it's partitioned off by a yolk membrane or yolk sac. You have the embryo that almost always kind of sits on top of the egg, just like it does in the other reptile groups like birds. I don't know, well, you probably didn't do this. This is kind of an old school thing. In developmental anatomy, did you guys look at slides of embryos at different stages of development?
No.
No, because it's mostly, the way they teach developmental biology now is mostly a molecular biology class, so you don't pay attention to the gross anatomy of developing embryos. When I took embryology as an undergraduate, we spent a lot of time looking at chick embryos that were at different stages of development. And the nice thing about chicks is that the embryo kind of floats up on top of the yolk, as opposed to in amphibians where the yolk is pretty much spread throughout the egg, and when the embryo divides, it divides all the way through the yolk, which is a different way of cell division than when you have a plate of cells floating on top of the yolk, which is what you find in reptiles for the most part. So in this case, the yolk is separated from the wall of the female reproductive tract, and it's this kind of self-contained unit. The yolk supplies the embryo. The embryo is separated from the outer world by the chorioallantoic membrane and the outer shell. But in viviparous species, that outer shell is basically gone. The chorioallantoic membrane is still present, but then those membranes are essentially just pressed up against the uterine lining of the animal. And typically at one side of the egg, typically on the yolk side, you basically have the uterine lining becoming what we call hypertrophied. It starts to grow. It increases the thickness of the epithelial layers of the uterus. And at that point, it begins to make these connections that move it towards a placental sort of arrangement, where this hypertrophied epithelial cells have a blood supply, and that blood supply then begins to provision the developing embryo. As a result of this, viviparous species tend to invest less in yolk when they create the eggs, because they're going to be supplementing whatever is in the yolk that is providing nutrients and energy for the embryo. They're going to be supplementing it with inputs from the uterus. And so this will then be retained inside the female until the animal is ready to be born through live birth, as opposed to this will be retained for a shorter period of time and will be laid outside, and a lot of the development of the embryo will go on outside of the female. So we've already talked about kind of this transition from oviparity to viviparity, and that transition oftentimes begins, you begin down that path of evolving into a viviparous species by retaining eggs longer and longer in the reproductive tract as a female. What are some of the reasons why females, if you're a reptile now rather than an amphibian, what are some reasons why females might, why a species might go down the path of retaining eggs for longer periods of time in the uterus?
So they want them to develop longer?
Okay, so yeah, but that's the kind of obvious reason. The question I'm asking is kind of what environmentally would make that advantageous? So maybe I didn't phrase the question very well. What is the under what conditions would it be advantageous for the egg to be retained inside the female? Because remember, there are costs and benefits to viviparity. One of the costs of viviparity is that the female is stuck carrying around these offspring for their entire embryonic development. So if those are significant costs, then what would the benefits be of retaining these eggs for longer and longer periods of time? Under what conditions would this be advantageous? Ecological conditions. Anybody? In the case of amphibians, under what conditions do females tend to retain eggs for longer periods of time?
The chance of survival, like of the eggs, is low.
In what type of environments?
For amphibians, it would be like if there's not a lot of water or a drought, I guess.
Okay, so it could be that there's simply a lack of a water source. The other reason that the survival of free-living larvae might not be very good is if laying your eggs in water and then having them hatch out in water means that they're prone to predation more in an aquatic environment than in a terrestrial environment. And so this movement from an aquatic environment to a terrestrial environment tends to start animals down the path to viviparity, because the longer the eggs are hanging out on land, not in an aquatic environment, the more likely they are to desiccation, whereas they're way less likely to desiccate if they're just retained by the female. And so a lot of times in terrestrially reproducing organisms, they tend more towards direct development or viviparity. So what's the environmental constraint that pushes reptiles towards viviparity? It isn't moisture, because they have a shelled egg and they should be able to lay their eggs in drier environments, because the added mineral layers of the shell will prevent those eggs from desiccating as easily. So it's not moisture. What's the other environmental constraint that reptiles are concerned about?
Is it temperature?
Temperature, yeah. So in many cases, organisms that occur at higher latitudes or at higher elevations, where temperature begins to impose a constraint on reproduction, you have females retaining eggs for longer and longer periods of time, because presumably if you lay your eggs under a rock someplace, even though that rock may have a stable environment, that stable environment may be a stable, cool environment rather than a stable, warm environment. Whereas retaining the eggs in the female, as the female thermoregulates, the eggs then take on the same temperature as the body temperature of the female. And so they're now being incubated at a higher temperature, because the female is actively thermoregulating. And as a result, they will develop more quickly and be more likely to go to full development, simply because they're in a warmer environment. And the environment in this case is actually inside the female. Once you kind of go down that path, if you're retaining your eggs for longer and longer periods of time, there's no need to continue investing in a hard shell, because they're not really going to need that hard shell for physical protection or to avoid desiccation. And so you get rid of the shell. As soon as you get rid of the shell, now you have these embryos in an egg with a membrane around it up against the placental wall. And then that just kind of sets the stage for then the development of a placenta. And then you're basically on the path towards viviparity. So your book talks a lot about kind of these ecological correlates. And not all organisms, not all reptiles that live at high elevations are viviparous, but a lot of the reptiles that are viviparous do live at high elevations or at high latitudes where they have to contend with cold. So it's one of those rules where not all high elevation lizards are viviparous, but almost all viviparous lizards live at high elevations. One of those types of situations. So there are hormones involved in this transition to viviparity. Progesterone, which is a hormone that you guys have, is also present in reptiles. It leads to the production of corpus luteum. The corpus luteum is this mass that it becomes its own hormone creating structure, and it creates hormones that basically prevent ovulation, prevent the developing of new follicles in the ovary. And that then slows down the rate of reproduction. You produce fewer clutches because you arrest your reproductive cycle while you're incubating your current set of eggs. You have these thin eggshells, or in the case of true viviparity, a lack of eggshells. You get this vascularization of the chorioallantois, so this vascularization of this membrane here, and then you get the hypertrophy of these epithelial cells and the formation, in some cases, of this rudimentary placenta. So, as I mentioned, this is there's this general gradual evolution, increasing egg retention times, short developmental periods, thin eggshells, and the ability to extend egg retention without suffering developmental abnormalities in the offspring. Oftentimes, generally, the female will produce fewer eggs and the offspring will be smaller when you are moving towards viviparity, because there become spatial constraints inside the female as well, in terms of how many offspring and how big those offspring can be. So these are all changes that occur kind of in concert with one another as you move from oviparity to viviparity. So we've already talked about latitude and altitude as one of the main drivers of this. Females can incubate eggs at temperatures that are optimal for embryonic development, and it's facilitated by retaining the eggs inside the female because the female is doing active thermoregulation. This is a picture of a thermal gradient. We talked about how you measure preferred temperatures in organisms, and I drew for you on the board kind of what a thermal gradient looks like, but this is an actual thermal gradient that we used to look at preferred temperatures in geckos a couple of years ago, and it's basically just a series of channels, and they're covered with hardware cloth, and at one end of the room, this is a very cold refrigerated room down in the basement on the ground floor of White Science. And what we do is we cool this room down so that on the right-hand side here, there's no heat lamps. On the left-hand side, there are heat lamps. And this basically sets up a gradient of temperature from about 105 degrees Fahrenheit on the warm end up here, and about 50 degrees Fahrenheit on the cold end down here. And you can see that there's a lizard right here, there's a lizard here, there's a lizard here, there's another lizard here, there's a lizard here. They're all kind of hanging out in the middle of this gradient. They're not on the very, very cold end. They're not on the very, very hot end. Well, one of the things that we see in Mediterranean geckos, Mediterranean gecko is a oviparous species. It lays hard-shelled eggs. But one of the things that we see is in gravid females, when they have eggs that are in development, they will tend to hang out at slightly higher temperatures than organisms that are non-reproductive. And so I'm not sure which of these is reproductive or not, but this lizard here and this lizard here, they're closer to the heat lamp, so they would be thermoregulating at a higher temperature. This lizard here, this lizard here, this lizard here are further back in the thermal gradient. They're thermoregulating at a lower temperature. But the thing about a thermal gradient is the lizard is free to move up and down this channel. When it gets too cold, it can move closer to the light. When it gets too hot, it can move further away from the light. And the way you measure temperature preference is you just come in periodically and you measure what the actual body temperature of these individual lizards is over the course of about a 4 or 5 hour period. And over the course of that time, you get a pretty good idea of what an individual lizard's preferences are, but then you also have a measure of what the overall population's preferences are. And so one of the things that people have found with lizards and snakes incubating eggs inside their bodies, whether they're oviparous or viviparous, is they tend to thermoregulate at higher temperatures. And you can measure this by just sticking gravid and non-gravid females in a thermal gradient and measure what temperatures they're actively thermoregulating at. Questions about oviparity and viviparity? Okay, parental care is found throughout the reptiles, and it runs the entire spectrum from absolutely no parental care to quite extensive parental care. Among the groups that have the most extensive parental care are the alligators and crocodiles, the crocodilians. In the case of crocodilians, they tend to build a nest. The nest is oftentimes formed from mud and vegetation. And if you're ever in a swamp, which I know some of you will never be, you'll come across occasionally in the swamp a depression, and then there's a mound in the middle of the depression, and then there's another depression, and then regular land on either side. And this depression, if you're ever walking through the Everglades in Florida and you come upon one of these, you should leave immediately because you've stumbled across an alligator or crocodile nest. And what has happened is the female has gone in here and she's dug a trench that goes around this mound in a circle, And she's dug that trench and she's taken the stuff that she's dug up and piled it up into this mound. And then she digs down into that mound and she lays her eggs. And then she covers the eggs up. And the mound does a couple of things for the eggs. Is it protects them from some predators, although raccoons will totally dig up an alligator nest and eat the eggs from it. But the main thing that it does is with all of this wet vegetation and mud, it starts to decompose, and the process of decomposition creates heat, and so it becomes kind of an incubation chamber for the developing eggs. And what the female will do is she will occasionally dig down into her nest, stick her snout into the nest, and there are receptors inside the nose of the alligator that allow her to determine what the temperature is inside the nest. So she has thermal receptors. And if the nest is too hot, she will take layers off of the mound. And if the nest is too cold, she will put layers onto the mound, and she will come and check the mound repeatedly throughout the day to monitor its temperature. Once the offspring have hatched, they're buried down in this mound, and they start making these little chirping noises, and the female will hear those chirping noises through the dirt in the mound, and she will come and she will dig those offspring up and liberate them from the mound. And then for a period of time after that, the young offspring will travel around with the mother. You'll have seen pictures of alligators with babies sitting on their heads, sitting on their backs, et cetera, et cetera. And so there's this extended time during the young life of an alligator or crocodile when they receive protection from predators by basically just hanging out with the mother until they reach a particular size. So parental care can be quite extensive. There is a group of birds that occur in Australia. They're called mound building birds, and they do a set of behaviors that are surprisingly similar to alligators and crocodiles. In the Mulga region of Australia, they will build these big mounds made up of, once again, dirt and vegetation, and they will, throughout the day, dig little holes in the mound. They'll stick their head in there. In their nostrils, they also have temperature receptors. They gauge the temperature of the mound, and they will once again remove layers if it's too hot and add layers on if it is too cold. And so one of these examples of convergent evolution, in this case in a small group of species of birds that live in Australia and alligators and crocodiles worldwide, but strikingly similar behaviors. We also see parental care in pit vipers. And so here you can see a rattlesnake, a New World species. This is the female, the adult female. This is her head right here. And you see that she has a couple of babies with her. They are viviparous, they give live birth, and there is a time period during which the young offspring will once again hang out in close proximity to the female. And during this time, presumably, they're getting the benefit of the female being able to defend them from predators and whatnot. You'll notice that in both these cases, these little babies are about to shed. And oftentimes, one of the first things that a baby snake does after it's born is it sheds off its most recent layer of skin. And so given that both of these things are on the verge of shedding, you can see that because of the gray eyes, the spectacle is cloudy. These may very well be newborn offspring. Parental care can also be undertaken by species that lay eggs. And so when you lay an egg clutch, the females or the males may stand around and guard the eggs once again until the eggs hatch. This would be a more rudimentary form of parental care because it's just guarding the eggs. You're not necessarily doing anything to help the eggs thermoregulate at a particular temperature, but you are presumably protecting them from physical damage and specifically protecting them from predation. So when we think about the anatomy of reptiles, reptiles are constrained by the fact that they lay a shelled egg. The presence of the shelled egg means that there has to be internal fertilization. So there has to be some mechanism for getting sperm out of the male and into the female. And in the case of crocodiles and turtles, there is an intromittent organ called a penis. In the squamata, in the snakes and lizards, you have a hemi penis, and in the rhynchocephalians, the tuataras, you have a rudimentary hemi penis. And so all of these are structures that are involved in transmitting sperm from the male into the female. Internal fertilization then occurs, and it's only after an egg has been fertilized that it then develops a shell. It goes through a structure called the shell gland, and a shell is applied to the outer layers of the already fertilized egg. So specialized anatomy that is not found in the amphibians because of the constraint of the shelled egg. So just like when we were talking about reproduction in amphibians, you need to get in the habit of thinking about all of these things as having potential costs and benefits. Shelling your egg has a lot of benefits, less prone to physical damage, less prone to desiccation, but then that implies a set of constraints where you have to somehow get that egg fertilized before it comes out of the female. And so there are trade-offs to be made in all of these kind of solutions to the environmental challenges of, in this case, reproduction. Now, in some groups, there are other constraints, and these constraints impose upon the arrangement of organs within the body, and also impose upon the arrangement of reproduction inside the body. So especially in the case of snakes, these long bodies of snakes place a lot of constraints on kind of how the internal organs are arranged. And one of the things that happens with snakes is that they have lung reduction. So they essentially rely on only a single lung, and the other lung is reduced in size. So in the case of snakes, it's the left lung that is reduced in size. So here we have a generalized snake. And there are two parts of the lung. There is a vascular lung that is here at the front of the body. And then there is a saccular lung that is here at the back portion of the body. The vascular lung is the lung that is actually involved in exchanging gas, and the saccular lung is just involved in ventilation of air into and out of the snake. But you can see that in many cases, the lung itself takes up 2/3 to 3/4 of the body length of an individual, which means that then other organs are relegated to the rear of the organism. So you have testes that are located posterior to the lungs and the small intestine. These testes are very long and thin, just as ovaries would be long and thin. The kidneys are also long and thin. So all of these structures are just essentially elongated in snakes, and the same would be true. This happens to be a male, but the same would also be true in females. And then what you have up here is the specialized anatomy of the squamates. These are the hemi penises of a given snake, and they're paired structures. And this is when they're everted. One of the ways that you can tell if you have a male or a female snake or lizard is you can gently depress the tail right past the cloacal vent right here. And when you press here, what the snake will basically do is the snake will evert its hemi penises, and you will be able to see them. And if it's capable of doing that, you have a male, obviously. And if it's incapable of doing that, you have a female. But these are basically like a glove. And so let me get a glove. You can think of the hemipenes as a hollow pouch, and they're inside the base of the tail. So imagine that my wrist is the cloacal vent. So the place where the cloaca is, where the anal scale is. And when the hemipenes are everted, what basically happens is the hemipenes basically turn themselves inside out. And when they come out, they appear like this. And so imagine that these are the forked portions of the hemipenes. And so it really is just this hollow pouch that then gets turned inside out of itself as it emerges from the snake, or the lizard for that matter. And so when they are inside the tail, let me recreate my hemipene made out of a rubber glove. If I can, maybe I should just get another one. Would be faster. So when the hemipene is inside the snake, it basically forms a hollow pouch inside the tail of the snake. So another way in which people will test to see if you have a male or a female is you can go to the anal vent where the anal scale is. You can take a blunt object like a bobby pin that you've straightened out, and you can insert it into the base of the tail. And if that bobby pin inserts deeply into the tail, you've basically probed into the pouch that is the hemipenis. And if it only goes a short distance in, you have a female because the female lacks the hemipenis. Both of these methods of sexing a snake have difficulties associated with them. I've seen people try and depress the tail of a snake to get the hemipenes to evert and break the tail of the snake because they manhandled it too hard. I've seen people manage to avert the hemipenes of the snake, but they press so hard that they've done something to the snake that prevents the snake from retracting those hemipenes in. So you can damage a snake, sexing a snake this way, or a lizard this way. I've also seen people probe a snake or a lizard, and once again, do it not gently enough such that they've punctured the hemipenis by probing too hard into the base of the tail. So I don't recommend either of these methods for sexing individuals. Either of them work if you know how to do them appropriately, and both of them can go terribly wrong if you don't know how to do them properly. They make special probes that you can buy from reptile collecting houses that are made for probing the hemipenis of snakes and lizards. I always found a straightened bobby pin to be just as effective and a fraction of the cost. But because the hemipenis is stored inside the base of the tail, oftentimes you can tell males and females simply by looking at the morphology of the base of the tail. You will generally see that females have tails that get very slender right after the anal vent, and males will have tails that are more thickened at the base of the tail because they possess those hemipenes. This is especially obvious in some lizards. The Hemidactylus tursicus lizards are very obviously male and female based on the fact that with the males you can see these bulges on the bottom of the tail that are due to the hemipenis. So those structures, these are the hemipenes, and they vary a lot by species, and so they're taxonomically important. And it's hard to see in this figure, but they're also barbed. And so when they insert these inside the female, they're covered in barbs, and this helps the male maintain contact, maintain the presence of the hemipenes inside the female, because these barbs basically, as the male places the hemipenis into the female, the hemipenis swells up, and these barbs go up against the wall of the cloaca of the female and kind of lock that hemipenis into place. And in some cases, copulation can be a fleeting sort of experience, but in some cases, copulation can be quite long, and the male needs to stay engaged during this entire time. And he comes with the armature to do that. Seems like a very unpleasant activity, sex in squamates, but that's the way it is. So those are the hemipenes. Embryonic development is pretty typical of most vertebrates. You have early cleavage stages, you have gastrulation, which is the formation of, well, blastulation, which is the formation of this hollow ball, and then the hollow ball begins to turn in on itself, and then you have the development of the neural crust that occurs fairly early in development. That leads to formation of things like the central nervous system and the vertebral column and things like that. And then over time, slowly, all of the organs are formed from these base germ layers. You have the ectoderm, the endoderm, and the mesoderm. The ectoderm gives rise to things like certain portions of the eyeballs, the central nervous system, the epidermis, dermis, those things. But then you have the endoderm, which gives rise to things like your gastrointestinal organs, your stomach, your spleen, all of those things. And then there is the mesoderm, which gives rise to muscle, and it's kind of a layered sort of structure where you have outer layers, middle layers, and inner layers in the embryology of all of this. So this is not a reptile embryo, but it's a chick embryo. But for those of you who did not take developmental biology back when it was known as embryology, this is what a chick would look like after, I think, 24 hours of development. And what you can see is, even at this young stage of development, it is undergoing quite a lot of differentiation. You can see that this is the development of the nervous system, which is one of the first things that occurs. The beginnings of a rudimentary brain. You have the development of eye buds growing out from the brain. You have the latter portions of the brain that are developing here, and then you have basically the dorsal nerve cord. Adjacent to this dorsal nerve cord, you have these regions of, once again, ectoderm that are basically going to form the skeletal portions of the protective, the vertebral column that's going to grow around this nerve cord. And then you can also see that there's a development relatively early on of a rudimentary heart. At this point, it's just a large thickened blood vessel, but it will eventually curl around on itself and form the four-chambered heart with an incomplete septum the four-chambered heart with a complete septum in birds and crocodiles, but an incomplete septum in the other reptile groups. And this is all done on a flat disc of germinative layer that is floating on top of the yolk. So cleavage in reptiles generally doesn't cleave all the way through the yolk. It cleaves through a floating mat of cells on the surface of the yolk. So the embryo is kind of floating on this big ball of yolk. And that's true of birds, and it's also true of the reptiles as well, the other reptiles, non-avian reptiles. Over time, you can see, so this is actually a chicken egg that has been opened up, and you can see that this is all the germinative layers that are floating on top of the yolk. And it has now created a very extensive circulatory system that is bringing materials from the yolk to the developing embryo. This developing embryo starts off by basically growing up from this layer of cells that, once again, is sitting on top of the yolk. And it will get to a point where it has grown up so much that it kind of flops over on its side. So in this case, it's still growing up vertically. It's just a ridge on top of this layer of cells. And as it develops further, it has grown up so far above that surface of cells that has kind of flopped over and lays on its side. You can see the beginnings of a tail developing here, and it looks almost like you can see the beginnings of some limb buds coming out. Oh yeah, this is a closer view. Once again, you can begin to see the development of the eye. You can see these clefts in the pharynx that are basically the rudimentary or vestigial gill slits. You can see in this case, because this is a chick, not a lizard, there are a lot of images that exist for developing birds. There are very few images that exist for developing reptiles. But this is the forelimb buds. These are the wing buds. These buds of tissue will ultimately come out and form the forelimbs, the forelimbs, which are wings in this case. And then there are buds of tissue developing at the back, which will ultimately become the hind limbs. And once again, you can see the spinal cord going down the length of the animal, and you can once again see these segmented areas of tissue that basically make up the internal segmented structure of the organism that becomes the trunk musculature and the vertebral column and all of that. So you can see this segmentation developing relatively early on. At this point, the heart is becoming much more developed than it was before, beginning to do this folding back on itself to form the ultimate force-structured type of arrangement. And so embryologists who study amphibians and reptiles will actually stage individuals. We didn't talk about this much when we talked about amphibians. We didn't talk about it at all, but there's a staging system for amphibian larvae. They're divided up into what are called Gossamer stages, named after a guy named Gossamer who developed the system. And so you can look at any publication where people are describing embryos, and when they describe embryos, they will tell you for a particular image what Gossamer stage that embryo is at, and then they're comparable across a variety of different species. And the Gossamer stage that you're at is dependent upon what types of morphological structures have developed at that particular point. And so there's also a staging system for reptiles as well, that once again are based on the different structures that are visible in the developing embryo. And these somites, once again, are going to become the musculature surrounding the trunk, and also you have ectodermal layers that are going to be right adjacent to those that are going to ultimately form the vertebra. You don't need to know anything about this staging system except that it exists, and it's based on the appearance of different structures. If you ever want to be a reptile developmental biologist, you will become well versed in these things. And then here you can see the limb buds, the forelimb buds and the hind limb buds developing in a later stage embryo. A couple of things I want you to be aware of is the development of limbs and digits. Generally what happens is you begin to have a limb bud, which is this outgrowth of mesoderm covered by ectodermal aspects. And then what you get is this limb bud will become longer and longer, and as it develops, it then begins to go through a process of programmed cell death. And so you end up getting a paddle, and then the digits themselves develop from this paddle by basically programmed cell death of the cells that basically become the spaces between the digits. And this is a pretty common pattern for development in all vertebrates. It's also how your digits develop as well. And so for those of you who study programmed cell death, apoptosis, one of the applications of studying that is in development, knowing how limbs develop, because that's through a process of programmed cell death. And that's, I think, all I wanted to talk about from the standpoint of embryology. You could take an entire semester-long course just in embryology. questions about this? All right, so just as amphibians have reproductive cycles, reptiles also have reproductive cycles, and there's a misspelling. This should be disassociated, not coded. Dissociated. All right. So in many cases, there is seasonality associated with reproduction in reptiles. And there are both prenuptial gonadal cycles and postnuptial gonadal cycles. And so nuptial refers to copulation, essentially. So we talked about nuptial pads or nuptial excrescences in certain amphibians, in particular the newts. Nuptial basically refers to the act of copulation. So there are cycles that happen with the gonads that are that occur before copulation, and then things that happen to the gonads after copulation. And the onset of reproduction, in almost all cases, has a certain seasonality to it. So if you can think about this in terms of the timing of the formation of gametes and mating, then when embryos are developing, and then when actually things like egg laying occurs. And so we can think about reproduction being continuous or acontinuous, or discontinuous. And discontinuous basically is when there are breaks between these reproductive bouts. And so you have gametogenesis and mating, and then embryogenesis, and then there's a period of time during which there is no reproductive activity, and then you have another bout of reproduction, and then a long break and a bout of reproduction. You can also have continuous reproduction, where there are very short breaks or no breaks in between reproductive bouts. And then you can have situations where you begin the process of reproduction, and then there is developmental arrest. So you continue carrying the offspring, but the offspring are not really developing, and they can stay there for a long period of time. And this usually happens in species where mating conditions occur at one time of the year, and conditions suitable for laying eggs or birthing offspring occur at a different type of year. Similarly analogous to plectrodonid salamanders, where courtship occurs most of the time in water, but egg laying occurs on land. And so discontinuous versus continuous, and both of these can be seasonally induced. Depending on where you live, the seasonality may be induced by something like rainfall, or it could just be triggered by things like day length and temperature. So right now, you are seeing a lot of movement of reptiles. We've been seeing the movement of amphibians for a while now. The first amphibians that I heard calling were Boreal chorus frogs back on March 11th. And so we're now at April 19th. So frogs have been calling for a while now. The first things that you will hear are the chorus frogs. They're always the first ones to start calling. Pseudacris maculata. A couple of weeks after those guys started calling, I started hearing occasional intermittent Acorus blanchardii, the cricket frog, and then over the last couple of weeks, I've started to hear Bufo americanus. calling, and also we heard that when we were out at Cooley Lake on Thursday of last week, it's the first time that I heard oh my gosh, I've forgotten Dendrophytes. I've forgotten the new genus of the tree frogs. That's why I hate it when people change genera. Anyway, the gray tree frog. Used to be Hyla versicolor, Hyla chrysosceles. I heard those calling for the first time on Thursday when we were out at Cooley Lake. And this is a pretty standard progression. You see some species coming out earlier than other species. But all of these species have come out basically because they're relatively cold tolerant, but they generally have waited until March when the possibility of really, really cold weather has declined somewhat. Unfortunately for these species, it's going to get cold tonight and it's going to snow by tomorrow morning, if you believe the weather people. And so one of the tricks with seasonality and things like temperature and day length triggering above ground movement and triggering reproduction is that weather is fickle sometimes. And in this case, it may be really bad for these frogs that have emerged at their kind of normal time when they're about to get hit by a period of relatively cold weather, maybe getting down into the mid-20s overnight, which many amphibians can handle, but reptiles can't necessarily handle those temperatures as well as amphibians can. And so the seasonality, when it's determined by something like temperature, is based on a long period of evolutionary time, evolving these seasonal cycles, such that it's a good long-term time to emerge from your burrows and start the process of reproduction. And even though that's a good long-term strategy, this particular year may not be particularly conducive to that. And so think of the seasonality of the onset of movement and the onset of reproduction as kind of a long-term average of climate and a species evolving to cope with that long-term climate. And as a result, that emergence time and reproductive time period may not fit any given year particularly well, but over a course of a number of years, it kind of averages out. It's going to be a rough night, I think, for some species here in Missouri. We're almost out of time, so let's talk about reproductive output, size number trade-off, and stuff like that. We'll talk about that on Wednesday, and then we'll probably move on from life histories to locomotion on Wednesday as well, because we don't really have that much left to talk about in terms of life histories of reptiles. Questions?
I have a question about the dichotomous key. Are we still planning
yeah.
on having to do Thursday?
I am, because I am working on those right now. I had some grading that I needed to get done in stats, so I kind of set down your dichotomous keys, but that is the thing that I'm going to apply myself to the rest of today. So my hope is to have them back to you in the next 24 hours, if not sooner.
Okay.
Yep. All right. Then I will see you guys on Wednesday. Rose, I have a question for you. If you could hang out for a moment.